in automation, we want to standardize what we are using. We don't want, 10 different racks sitting in the lab and, making automation harder. So if you want to have, customized rack and a standardized rack for all the applications, then 3D printing would play a huge role. We can as well use 3D printing for mounts for electronic components, or we can design our own custom tube connectors in various configurations as needed by the project. And as explained earlier, with 3D printing, we can work on chemically compatible materials. That's the biggest advantage, in a chemistry or biology lab. So here are the examples of some custom solutions which I designed. We have racks for vials with different diameters and in the same format as an SBS rack, because that's the format which is being used in most of the machines. So this is one of the examples. So this example will, help us in standardizing the racks for automation purposes. But, we can also design something like this, singular vial holder. The micro vial have, they have a round bottom or, round bottom flask. There's no way we can make them stand, right? So we can as well print a singular vial holder or gather them together into a modular rack in the same SBS format. So that's the utility of 3D printing. Or we can have some novel solutions. Alex explained yesterday, we are using SP. It's a solid phase extraction method, and we designed a manual station as well as an automated station. Both of these stations have a lot of 3D printed parts. For example, the sealing plug is made. It's made out of an elastomeric 3D printing filament. It's thermoplastic polyurethane. For the sealing plug, it's wetted parts, so it's made out of PEEK because it's the most chemically inert plastic we have. The racks here are made out of polypropylene. They too are 3D printed. The cartridge adapters here, the model cartridge adapters, they also are 3D printed. So there are a lot of 3D printed components to this. Then there's another solution for vial evaporation. The solvent evaporation in a vial. So we already have some existing bulk equipments in the market, but all of them do like 100 vials at a time. But we don't do evaporation 100 vials at a time. So I designed something which can be used for a single vial. The cap is 3D printed. There's an inlet for compressed gas. There's an outlet for evaporated solvent. It's compact, as you can see. It's benchtop compatible. You can just use it in the hood as this is being used. I'll just demonstrate the working of this. So, it's creating a vortex, and as you can see, there's some condensation happening on the outside of the vial. Let's go with the next slide. So as I explained, 3D printing is helping us make rapid iterations to the design. So I'll just explain a few examples how that works, So for the benchtop vial evaporator, this is a lid, right? So I'll explain the design process which I used in this. The first step for me was to 3D print the threads, because that's the most important part for a lid. So once that was done, I had to print like a couple of prototypes to get that right. Once that was done, the next step was to design this helical system for a compressed inlet for, nitrogen or air. And that was my second step. The third step was to add threaded connections for tubing connectors. All of these were 3D printed. Like these are the computer generated designs in SolidWorks. And then all of these were 3D printed one by one. So, and then, this was the final design for this, this model. It has a screwed connection. It has 2 connections for tubing. But then, in the end, I tested these design with a lot of chemists in the analytical chemistry lab, and they found out that the threading was not working out. So I had to change the design, and Eric Walgren, he suggested me to go ahead and design something with an O-ring on top of it. So I just made some changes to the bottom part of the lid and added an O-ring. But then here comes one of the important parts. While iterating designs, I print them, give them to you guys, and then you guys provide me a valuable feedback on is it aesthetically good? Is it user friendly? And this was one small but important addition to the design. The lip on top of the lid, it makes taking off the lid easier. And this one step, if you use traditional manufacturing, you give it some outside vendor, and he'll take a week to do that. But with 3D printing, I just made this change in the computer generated design, and I printed it in a couple of hours for you guys. So that's how 3D printing helps us in rapid iterations. And then once this is done, we can scale it up for an automated platform. So this was the final prototype, which is being used by around 15 chemists in the lab. And they are providing me valuable feedback at each moment, so that I can make incremental changes and go back to the prototyping stage. The second project was to scale it up, as I discussed, right? So this was a single vial evaporator here, and then I made a batch evaporator. So firstly, I discussed with the chemists, and they told me they might need individual flow controls for all 6 vials. So that's what I designed. And this is the prototype, all of which is 3D printed. All the white parts are 3D printed, and these are the connectors which you guys already know about. So this worked pretty well. It could evaporate 6 vials at a time. But the thing was, we discussed, we brainstormed, and we came to a conclusion that we don't need individual flow controls. So there you go. The next step was removing all of them, having a single inlet and a single outlet. And this definitely made the design easier, and rapid manufacturing helped in that. I just made a small change, and voila, next day I had this design in hand, all of which is 3D printed. And then once the design was easier, I made a 2-tier system. All of these parts are 3D printed. All these modules, all these fixed parts are 3D printed. And this is fully 3D printed final iteration of a batch evaporator. And then Eric went up another step and added some more functionality. He added a heated plate and copper infused rubber holders, so that the vials can be heated while evaporation is being done. But the helical thing is still 3D printed. So that's how it works. We make prototypes using 3D printing, and once we are done with the final prototype, we manufacture it with traditional methods or a mixture of additive and suppletive manufacturing, and we get the final working thing for you guys. So 3D printing is useful in that way. Then there are some more examples of 3D printed items. We can print a bioreactor dispenser. If it's broken, I can print one for you guys. You don't need to order a metal one, which will take a couple of weeks to arrive, right? So I can make one for you guys. If you want to install a camera in one of the robotic systems, the bracket can be printed in a couple of hours for the camera. Plate nest can be printed, a bottle holder. Pepper asked me to print a tubing holder, to keep the tubing in warm water bath. So I printed this for him, which can hold, some 5 meters of tubing inside it. So, that's how 3D printing is useful. So, yeah, concluding my presentation. What are the uses of 3D printing? Rapid iterations are possible where incremental design changes are required. No dependence on any outside vendor, and it saves a lot of time and money. Money in terms of it's an additive manufacturing technique. You're not wasting any material. You're just building on top, like layer by layer. So there's a lot of material wastage, and you have flexibility with the materials you are using. With additive, you will use aluminum or steel, but with 3D printing, you can use various plastics which are chemically compatible, and yeah, we can have custom solutions to the problem. Some solutions, if we are making them compact, the design becomes so complicated that that the traditional manufacturing techniques don't work with them. For example, with the helix, which I designed for the evaporator, you cannot use CNC on that. So 3D printing helps in custom solutions. So all of this was done in collaboration with all the scientists at NCATS. They provided me valuable feedback at each step, and it helped me make incremental changes to the design and arrive at a final concluding design, right? So thank you all for your help, and I'll end my presentation, I guess.
Great. Well, that was great. I guess I can start with one question, and then if there's anybody else, I think you can either put in the chat and I can ask it, or if you're so bold, you can speak up. I had a question about kind of what are some of the properties of the printed materials. I mean, are they prone to like warping, or are they brittle? I mean, is that sort of thing, right? Because if you have a metal part, right, that's one of the advantages is it's going to have these physical properties. But if these are plastic, are they prone to break, or does it really depend on kind of the design and what material you use?
Yeah, that's a really good question, because that's mostly asked about, it's plastic, it's brittle, right? But then we have a lot of varieties of plastics. As I told you, there are like 200 compatible materials. So we can use polycarbonate, which is, not that brittle. We have metal infilled plastic, it has metal powder infused in the plastic. I have worked with copper infused plastic. There's wood infused plastic, so these guys, if you work on some mixture of plastics or some other plastics, like polycarbonate, we'll have glass infilled polycarbonate as well, so it will help us, get a stronger part. I talked about infill percentage, so that's the hollowness of the part. If you want to make it just a prototype, you can put it like 20% infill. But if you want a stronger part, will be like 100% infill. It will take like a couple more hours, right? Regarding warping, some materials are heat resistant, some are not. So with 3D printing, we have that flexibility of choosing the material which we want. Like if you want to focus on heat resistance, we'll choose one material. If you want to focus on chemical inertness, we'll choose another, right? For chemical inertness, we'll use PEEK, because all the connectors which you guys use are made out of PEEK, and PEEK also has a good thermal stability. So we'll use PEEK for that, right? So, and for warping, 3D printing can cause some warping while the part is being printed, but then we have so much flexibility with the settings of the printer that we can change the temperature at any layer, and we can work with all the parameters of printing and get as accurate part as possible. So that's that's all about rapid prototyping. I make one, and then I see the dimension is not correct, I'll make another in a couple of hours, and it's all a fast process, right?
Yeah, great. I guess a question's coming in from Dr. Iyer. The question is this service open to other NIH scientists who are looking for the custom product, or would this be something that we envision if it's not formally available? Is this something that do you plan that we could open this up, or are there other capabilities at other institutes that you know of?
So right now, it's open for NIH for sure. But right now, we have Ultimakers, which they don't work with PEEK per se. They'll work with other materials. So you can provide us a custom product or custom design, and we'll print it for you. So that's how it's been going right now. So I got a demand from a scientist from NEI, National Institute, and he asked me to print something for him with Ultem. But my printer doesn't do Ultem right now, so I got it done from an outside vendor, but I'll definitely help with that. For future stuff, we have a makerspace coming up in the new Aspire lab area, and we'll have more 3D printers like the one which can print PEEK or Ultem or some more varieties of materials. And that makerspace will be shared space for all of NIH scientists. And so the thing is, you guys will provide us the design or a project which we work on, and we'll work with you in collaboration with you guys, and get you the 3D printed parts.
That's great. So I guess contact they have our contact info if this is something that you want to follow up on. Anybody, just again email any one of us, and we can kind of put people in contact with the right people. Thank you so much for this great talk.
Sure. Yeah.
So just to kind of move things along now, we are going to now move to talk from Jamie Travers, who is a laboratory automation specialist at NCATS. And he's going to be talking about our Cobotics project, applying adaptive optimization techniques in cross-platform communication to enable informatics-driven experimentation. So.
All right, just make sure everybody can hear me first. Is that good?
Yep.
All right, share my screen. All right, can you see that?
Yep, we can see it.
Awesome. So I actually did an actual poster for this. It's not a presentation, but I'll be able to walk you all through it. So I'm just gonna jump in right here. At this point, you've heard a lot about Aspire, including the mention of our goal to achieve a chemistry-focused drug discovery workflow as a continuous and iterative process. For the last several decades, we've had far more biological screening capabilities, and the idea is for chemistry to kind of catch up. We want to show that we can apply steps to aim towards autonomy in the chemistry workflow, and this collaboration with Cobotics, which is a startup up in Boston, is one of the many simultaneous starting points we have for Aspire right now. This project spreads across the kind of what, how, and the make up in the diagram up at the top in regards to automated biological assay optimization, and it's kind of a proof of concept which will be applied to reaction chemistry. To that end, you can see on the top diagram that the assays and chemical libraries act as inputs to perform high throughput and virtual screens depicted by the green arrows. This data is analyzed to determine both optimal assay conditions and to generate dose response curves. And the resultant analysis is used to determine molecules to be synthesized, which are produced and then placed into the chemical library for further testing. These outputs are shown by the blue arrows in that diagram. And the overall figure kind of just displays the system of an autonomous assay execution and chemical synthesis that we're working on right now. We're showing the utilization of artificial intelligence and machine learning to perform a design of experiment automatically for biological assays. And in this case, we chose the papain assay, which is very well characterized and it's cheap enzyme. So we can kind of just figure everything out, run some experiments. If you look below in the results section, you'll see the assay table showing the steps of the protocol, including enzyme and substrate addition by a time 0 fluorescence read, followed by some incubation time and a second plate read. We used no enzyme in 2 columns as a control, and the parameters we decided to vary were enzyme concentration, substrate concentration, and incubation time, of which there were 8 of each for a total of 512 combinations. And before I get into the details of what we accomplished and the results, I want to cover some of the design philosophy before we do that. Before you can kind of think about integrating the virtual and physical interface, you need to have a way to send as well as receive messages. Advanced Messaging Queuing Protocol, or AMQP, provides a platform agnostic method for ensuring information is safely transported between applications, among organizations and within mobile infrastructures and across the cloud. So you need the cloud controller between the virtual brain and the physical system, essentially. We went with RabbitMQ, which is a means of implementation for AMQP protocol. RabbitMQ is the most widely deployed open source message broker. The NCATS RabbitMQ server has been placed in our DMZ, our network DMZ, such that Cobotics can connect to it and send messages without any issue and securely. We have validated the new server IP at NCATS and have worked with Cobotics to ensure connectivity. And without this messaging piece in place, we would not have been able to accomplish any of this. The design is based on command query response segregation, or CQRS. Our screening platform queries the Cobotics virtual, and the Cobotics virtual brain analyzes and sends commands back. We run all of our experiments using an event-driven system, which is based on resources available and not necessarily based on time. Another hugely important aspect is an asynchronous operation, meaning that multiple commands can be issued and multiple queries can be processing simultaneously without being time dependent. Each assay plate is its own independent experiment, and theoretically no limit to the number of assays that can be run concurrently on the system. So now we're going to take a look at the messaging workflow kind of shown right in the center there. We'll go over a little bit of the AMQP workflow. Start by looking at the brain, which represents the Cobotics and the AI machine learning platform that they have up in Boston. And we work with that in conjunction. The steps are also listed in the virtual and physical figure to the right of that messaging workflow. Kind of just gives you a little more of a, I don't know, a simpler, more layman's terms breakdown of what I'm going to kind of be going over on the left figure here. First and foremost, SAM programmed a safety indoor scheduling software for the robotic system where we need to acknowledge through pressing a button that we're ready to accept messages. Basically, essentially allowing it so that we have full control over our system, and it's essentially just for safety purposes. This allows us to ensure the system is ready and any setup is complete, including reagent setup, calibration, plate loading, etc., before we can begin. Step one is where a message is produced, routed through the exchange, and queued by the Cobotics virtual brain to run an assay with specified conditions, including one enzyme each and substrate concentration and an incubation time. The screening system then acts as the consumer, which is dequeuing that message sent from Cobotics, and it automatically creates a method with the optimized parameters sent from Cobotics. And the enzyme and substrate concentrations correlate to different tips on our dispensers. An assay is next launched using the method that was programmatically generated. And there's no limit on the number of concurrent assays where each plate is in its own independent experiment. The plate is then read with the resultant data being the message that is produced, routed through the exchange, and queued and sent to Cobotics. The data is then consumed by Cobotics, and the data is processed and analyzed. And now that we've arrived at kind of the last step, only to start it all over again, but here is where we use the processed data to update the AI machine learning model to generate new conditions to try. This is kind of an iterative protocol, and it continues until the desired or optimal conditions are met. So we're just going to continue along this loop until we essentially get to our optimized conditions. So that about covers like the main technical components. And again, the messaging protocol is what makes all of this possible. So now we can kind of dive into the actual optimization of both the papain assay as well as the optimizer, which needed to be created on the virtual side. This was accomplished by Sam Elder of the Cobotics. He developed the equation, which improved over time considering parameter weighting, rules, and cutoffs. There's an establishment of true objective function, and the inverse cost of Z prime is greater than 0.5. You can see an example of one such equation next to the green assay table. It's in this gray area right here. So that little bit of code is kind of what helped us. Oh, did I lose my presentation? Yeah. All right. So before we ran a brute force experiment on a main screening platform where we tested all 512 combinations across 5 days continuously and autonomously with absolutely no intervention. The 512 combinations were sent by the Cobotics platform in a randomized fashion. This was also running simultaneous to other projects that needed to be screened on the robot, basically just in parallel with our daily work running cell titer glow assays and kind of pretty much anything else anyone needed. Performing the brute force experiment served multiple purposes. It allowed us to test the robustness of our messaging protocol, and it generated simulation data that could be used by Cobotics to learn the model and would serve to prove that brute force not necessarily worth it when we compared it with using our automated assay optimizer. I'd also like to note that we also ran this process using plate washing. So instead of running 512 plates for each condition, we only used about 40. Basically, once plate got through its entire method, we would wash it with, I believe for mainly biochemical assays, we use ethanol and water, a combination of that along with some aspiration. So we're essentially only using 40 plates to run 512 conditions. So it saves us time and resources. The last step in the protocol is to aspirate the assay material and rinse the ethanol in water before being returned back to the queue for another round. The key point I want to make before moving on is that with solely brute force method, you're very limited in the amount of parameters you can vary without exponentially increasing your cost in runtime and complexity. So at some point you won't be able to add more, and it's not necessarily worth pursuing. But with combining the virtual and physical systems, we are essentially limitless in that regard. Next, I kind of want to summarize the 4 rounds of optimizers we ran, which took place after the simulation optimization that was done by Cabotics. This was run across 4 days with multiple processes running in the background for other projects. Again, the 4 scenarios we executed were ones where messages were sent slowly as well as fast, and each of these were combined with and without a log transform. We start with the randomized messages, and this includes the enzyme and substrate concentration as well as an incubation time as the variables. Once 5 randomized message assay results have been analyzed, then the optimized messages start to be sent in addition to random messages. These optimized messages are influenced by the process data to make a decision on what it thinks should be tried next. We're showing results in the orange and blue graphs, which are only from the second trial of optimizers, but we had 3 total where each time the equation was improved. Looking at the bar graphs where orange equals an optimized trial and blue equates to a random trial, you can see that the 2 scenarios perform better than the others. These were fast experiments combined with rescaled inputs and slow experiments combined with log inputs. Each time, near 70% of the time, the optimized messages yielded the NCAT subjective, which Sam at Cabotics defined as 1.4 or greater based on the developed model, taking into account what success looks like for us. Overall, though, taking all 4 scenarios we run, we saw 50% of the optimized trials reach or surpass the objective. This is clearly more efficient than running the brute force method. It takes part of 1 day to reach the optimal trial versus 5 days using the brute force method, and it also uses significantly less reagents and saves us money and resources. The third round of optimizers, which ran continuously across 3 days, added smart random messages and even lower budget for the trial number. It also incorporated dynamic compound addition once the optimized conditions were met. The Gantt chart is shown under the green assay table down at the bottom right there. And again, a mixture of random and optimized messages were sent. And upon meeting optimizer criteria, a message is sent to initiate compound addition. And you can see where the orange line shows this process kickoff to, like, there's kind of a splits the graph down there at the bottom. But the assay protocol using those optimized variable combinations along with an added step in the method for corresponding compound addition technique. This included both pin tool and acoustic transfer of known pathway inhibitors, as well as a pharmacologically active compound selection, all in the QHTS format. Finally, dose response curves were automatically generated by our informatics platform, and in which case this was done on Palantir on our NIDAP platform. But underneath the bar graphs, there are the 11-point dose response curves showing that we did indeed get the desired activity by those known pathway inhibitors. And to the left, we display a heat map of one of the assay plates where the Z prime was 0.7, which is pretty significantly high. This was a great success in building a necessary tool for the Aspire project. The goal will now be to design chemical compound optimizers to allow us to potentially find more potent and desired chemical structures to screen next from an existing source plate within the library, or to kick off the automated synthesis of a new small molecules to test. Yesterday, you heard Sam mention the Techmatic Indigo Mobile Reactor in California at the Lilly site, and you'll hear more from Mark Ding on his work with it today. We'd love to transfer this messaging technology to the Indigo reactor once NCATS has their automated integration with liquid handlers in Maryland for amenable chemical reactions to enable a fan-out experiment of sorts, since multiple Qs can be bound to the same exchange. This way we can optimize our reactions alongside each other, ensuring both the reactor in California and the one in Maryland yield the same output, to essentially increasing our reproducibility. And that's it for my presentation. Thanks for having me, and I guess I'll open up to questions, which I believe Jamie and Carlene will be helping field.
Yeah.
So thank you.
Sure. Thanks, Jamie.
Mhm.
Is there, if you have any questions, put them in the chat, or again, small enough group that, just unmute yourself. I'll wait a minute, and then, I have maybe some leading questions.
Sure, I can certainly try to answer them.
Well, I guess, one question you could ask is, the pain, this is a pretty simple assay, right? And it's really hard to screw it up. I guess the intent here is that this was proof of concept, and that we can apply this to more and more complex assays if we're talking about biology. Correct?
Yeah, absolutely. We kind of just chose this one for its simplicity, that, like you just said, basically as the proof of concept, because we know this assay works. It's very well characterized. We've done it here a bunch in years before this, and we know it works on our system. So it's naturally kind of just a good one to start with, for sure. And yeah, that's ideally, we're going to start working this up into more complex operations. But for right now, pat pain seems to do the trick.
And then I guess one thing to really reiterate is that the plan isn't just to have this for biology. It's, you mentioned the indigo reactor, that this can be used not just for designing assays, but it can be applied to designing optimal syntheses for particular compounds that you want, correct?
Yeah, for sure. Like, this is going to be, I would assume this is going to be heavily integrated into like the new Aspire system, and that we'll be able to have that kind of eventually have that kind of closed loop of just having everything all within a single system, that once we like determine like a lead compound or a hit compound on a specific assay, or determine like a theoretical compound, that we can synthesize all within the same system and send it back to the beginning for more testing and optimization. Yeah, that's the ultimate goal right there, is to kind of have everything all contained in a single system to be able to do it all.
There's a question here. Any future for label-free assays? Could this be applied, let's say, to optimizing a label-free assay? And I guess I would pivot that a little bit to just, any biological assay in general. Could you, in theory, use this technology?
Yeah, I would think so, because like it all comes down to like the messaging workflow. It's like, how can you modify? You don't even necessarily need to modify your assay to get in on this, because like we're just doing this with essentially just a pretty normal biochemical assay. So I would assume that we could definitely modify our workflow somehow to work with a label-free assay.
Yeah.
Of course, that would take some research.
And
yeah, please, Carlene, add something.
Yeah, and so we actually are, Thomas, we're integrating a RapidFlex, Bruker RapidFlex for mass spec onto our larger robotic system. So that is something we'll definitely be able to add. And yeah, so to come.
Yeah, and I'd also mention, just to pile it on here, that when you're talking about biology, one limitation is how stable are your reagents, right? And so
exactly
the applicability to a given optimizing a biology assay, that really is going to depend on how stable your reagents are, right? Because if you prepare it and you got 10 minutes to use it, then you're going to need somebody in there to be doing this more manually. But if you got something like pain where it's pretty stable through a whole workday or overnight, then you can do probably more extensive work with this. So again, like everything is like context dependent, right?
And Jamie, you bring up a great point, because throughout Aspire Day we've been hearing, well, we're going to have a fully automated system or lab, and that's not necessarily the goal or where we want to be or need to be, because these are tools that we use and we need people to set them up, right? So you have to have somebody making up these reagents. Like for this experiment, for proof of concept, we had, Adam Yasgar was making up the reagents. We are, refilling them as needed. And you're not going to take the person out of this equation. You could have a mobile robot thaw a reagent and make up an enzyme. I don't think so. I mean, you could, but
not yet. Not yet.
And, the idea isn't to take out the person, but the idea is to get everything set up into a way where you can have it continuously run. And again, we're not necessarily looking to do this for biochemical assay optimization. This was, as both Jamie's mentioned, this is proof of concept for, to apply to other endeavors. And that could be cell-based assays, that could be chemical synthesis. It has many possibilities.
Awesome. I think it's great. Thank you, Jamie. It's weird saying thank you, Jamie.
Yes.
Yeah, and great job, Jamie.
Oh, thank you.
Appreciate it. Thanks.
All right, thanks again. So we'll move along here now to our next talk by John Januszewski, who is a research scientist here at NCATS, who's going to be talking about something that's really exciting to a lot of us here that are interested in applying mass spec to whether it's biology or chemistry. So he's going to be talking about an open port sampling interface for high throughput mass spectrometry, emerging biotechnology for screening and reaction optimization in drug discovery. So John, it's really nice to have you, and take it away.
Okay, thanks, Jamie. Can you guys see me? See my slide?
Yep, see you, hear you. Great, thank you.
Okay, awesome. All right, so this talk is the work of Megev, Sam, and I over the past year in Catch-as-Catch-Can in getting this open port sampling interface to work, along with our mass spec. And so that's what we're doing. There we go. All right, so this essentially outlines the poster. I'm going to describe the open port sampling interface, the acoustic ejection mass spectrometry, and then an update on where we stand at NCATS on our OPSI MS platform, and kind of a current state, future state, where we are now and where we're going. All right, so this diagram depicts the open port sampling interface. It's essentially two coaxial tubes, a tube within a tube. At one end is a standard, for the most part, standard electrospray ionization tip with high pressure nitrogen gas coming in, and the sheath gas, nebulizing gas, creating a Venturi drag on the system such that solvent entering in the upper chamber here or at the sampling tip is sort of sucked into the inner tube and out to the mass spec. So what we out to the mass spec detector. So what we have here, essentially a flowing stream open to atmosphere. These dimensions are misleading. Typically, we're using a tip that's about 1/16 of an inch in diameter. And you can, for example, this is a flowing stream, right, open to atmosphere. So you can take a sample with a pipette and drop it here like a microliter and read the output on the mass spec. Now, this is the work of Gary Van Berkel's group at Oak Ridge National Labs, Bill Mos-Cortez, and they've really done some wonderful work, and there's a boatload of publications by Gary and his group out there, and this is one of them. This is a very simple system. This is really the magic of the system, simplicity. This is a surface sampling app where they've inverted the sampler. So this is the sampling tip. This is a very, by the way, a very standard HPLC T. So here is an HPLC fittings all around. here's our solvent coming in, going around the, down through the sampling tip and back up into the reader, into the mass spec reader. In this case, there's capillary, this is a capillary tubing going back into a valve. And in this system, they're not driving it by electrospray sheath gas flow. They're driving it by a syringe pump. So this is open, this is open for innovation. And we could certainly imagine this being coupled into a flow chemistry system where there's a valve. In this particular application, there's a valve in line separating the low pressure half of the system with the high pressure sampling half of the system. So it looks a bit like this. So this is the liquid junction of the surface sampler. And here's our solvent coming in, here's our surface. And what's happening, it's almost like a vacuuming over the surface of the sample or this, whatever you want to measure. All right, so this is also Van Berkel group. All right, one of the key features of this gadget is the physical shape of the tip itself. It can be tuned for different functional applications. So, for example, if you starve the tip of solvent, if you really crank up the drag on the system, you can starve the system and you won't get very good response, you'll get uneven response. So dial in the tip depending on the sampling tip, depending on what you want to do. Typically, we're using something that looks like this, where Gary calls it a critical vortex. It's basically a balance between the incoming flow and the outgoing flow to the mass spectrometer. You'll notice that the inner tip is recessed a bit from the outer tip, creating this sort of a pool here. And that pool can be utilized to create a dome or spillover mode. So this is potentially good for surface sampling applications and other applications. Very ingenious little gadget. So we're adapting this for acoustic ejection mass spectrometry, and this slide is from SciEx. So this depicts an acoustic transducer below a standard plate, and the red dots are our samples being ejected into the sampling tip through so here's our liquid coming in, here's our drag at the other end, our electrospray ionization tip. And here the samples processing through this transfer tube, high pressure gas being pumped in, creating a nebulizer, and being read as a standard electrospray stream. And this is this readout here. This signal is the generated signal from our sample. All right, so acoustic ejection mass spec, circa 2018. I want to point out that this is really emerging technology. Introduced on the market, I believe, in 2020. But this is the breadboard, and this is the kind of early stage here, and this is the transducer underneath a standard 384 well shallow well plate. Here's open port sampling interface. Here's the mass spec behind, and there's just put on an XY stage over the transducer and sampling well to well. This work was done at Pfizer by Wei Zhang and his group. And I want to just quickly, this is another recent publication, 2020 publication from Beringer, a group led by Andreas Leupold. And I have a short video here because this is really a picture's worth 1,000 words type of thing. So I hope you guys can see the sampler.
Yep.
Okay, so here is this is an EDC ATS 100. They're very common at NCATs. This is the SciEx mass spectrometer. This is the ion source. And I'm going to just kick this thing off. So this is the implementation. This is the setup we are going for. We have at NCATs. And that's literally this is how fast it's going. These guys, their claim to fame in this publication is going at 6 seconds per sample. So that what you're looking at is the sample readout, the signal readout. An interesting thing to note here is they're monitoring two transitions at 15 milliseconds per transition. So pretty standard stuff, but very fast. And they're at 6 seconds per sample. You're going through a 384 well plate in about a minute. All right, so that is what we are aspiring to do here. You'll notice that this is a very short transfer line. This is really a cooked system, a very well designed and innovative approach to modifying the EDC for sampling. And that's what we're aiming to do at NCATS. Okay, so to transition to our system, this is what our current workstation looks like. This is an EDC on a mobile cart. We can pull this is up, by the way, up in Crystal Clare in Ding Yin's lab at NCATS. We can pull the system around when we want to use it to our mass spec, and we can easily roll it out of the way when not being used. And this also on the left here is the EDC setup at NCATS. This is a 3D printed jig holding the open port sampling interface. This is our transfer line. It's 20 centimeters. These features are important to when you start thinking about the way you're controlling the system by the Venturi drag, the degree of drag on the system, and the flow rate into the system. So not quite as neat, but it works. So here, just for comparisons purposes, here's the Behringer system where they've taken the housing off of the ion source. They've cut out a piece of the deck. They've really worked the EDC, modified the EDC to allow them to use a 10 centimeter length of tubing here, which really starts and flow rates up to 1 mil a minute, which really increases the throughput quite a bit. All right, so here's our initial results. This is work that we did about a month ago now, 80 peaks. We're going at 2 seconds per sample. Our samples are diluted in 50% methanol. We have a 20 nanoliter ejection volume, and our coefficient, our CV is 11%. So we need to improve our precision, but we're getting very good separation between our ejected samples, right? So, and the flow rate here is 360 microliters a minute, 100% methanol. Okay, this is a standard curve we set up going well to well. There's a well, I'm not sure how Sam set up the EDC to do the sampling, but we put an empty well in between each set of the ejections, 20, n equals 20 for each concentrations, with bisphorone running from 0.03 to 1 micromolar. This is very, fairly good ionizer, good ionization efficiency, but we're not breathing that hard, and we have some work to do on our precision. But we have a good R squared here, taking the mean of these groupings for the standard curve. And this is just kind of a close-up of our low end, showing really nice signal and really good separation. I want to point out that the peak width at the base here is 0.9 seconds. The separation base to base is 1.3 seconds. So we have plenty of room to add another sample and essentially double our throughput. So that'll be something we're wanting to work on by increasing the flow rate. So we have some work to do here, but we're in pretty good shape in our current state. Okay, so ejection precision has been 10 to 12% at 10 to 20 nanoliter volume. We're running at about 2 seconds per sample currently, less than 1 second peak width at base. So this is just, these are figures of merit that tell us that the system is operating well. We need to reduce our sampling variability. Less than 5% RSD has been routinely achieved by many workers in the literature. And we need to reduce the ejection volume. 5 nanoliters is reported as a sweet spot. Work on alignment of our acoustic transducer to the open port sampling interface, and then run real samples. So I just want to point out that a lot of publications have come out within the last year. This is another publication from the Pfizer group on parallel medicinal chemistry, reaction optimization. This is the BI poster, Bristol Myers publication. A nice piece of work by Cheng Liu and the SciEx group on operational modes of the system. This is an interesting publication from Merck, where they did a lipid extract and they left the 2 phases, an aqueous and organic phase, in the well and acoustically dispensed through the phases to measure their diacylglycerol in this case. And this is a compound management application, SLAS technology. All right, so we have the system under control, acoustic dispense. We know what we want to do. We have some work to do, but we think we're in pretty good shape. What we'd like to do, and what Magev is working on, is developing this OPSI MS workbench. So this was developed, this is a known quantity at NCATS, it was developed at NCATS. We're working on a multi-function test bed for sampling with this open port sampling interface. It's a 2x3 foot deck. The arm, the macadamic arm, is like the key feature of the deck. It can be outfitted with a sampling tip for surface sampling. So we really want a scratch pad for the most part to test various ways of using the open port sampling interface for direct mass spec sampling. So this is just showing that jig of the OPSI. So we can put this, the workbench close enough to the mass spec to sample directly to the open ports, to the ion source of the mass spec. This is a Z drive dispenser. We're working on a pumping system. We want to, there's a lot of innovation still to happen on this OPSI for drug discovery, more so than, a lot of work's been done, but this is really early. I want to emphasize this is early days. So we're looking forward to working on the system. So this is a close-up of the OPSI in that jig, just showing that we can do a non-acoustic aspiration, dispense, and infusion sampling with this gadget. So this is just showing our liquid handling tip in relation to the inlet itself. I want to point out here that you can create a liquid junction by dispensing at low flow and balancing the dispense flow rate with the sampling rate of the OPSI. So we can achieve a really stable ion current. And why that's a good thing is you can imagine doing experiments on, integrated with the mass spec. So this is a mass spec readout. And the thing that is happening here is just the ability to create the stable flow. And you can imagine a intelligent system where and this is kind of common in mass spec field where there's, intelligent monitoring of the system. So if you start an experiment and you get the readout you want, if you see the fragment or the mass that you want, you can tell the mass spec, okay, change the experiment and do this experiment next, and so on and so forth. So there's many variations. You can use a differential mobility spectrometry to filter out certain classes of molecules on the fly. So you can imagine we're using different mass spec sampling technologies to sample one mass range here, another mass range here in various ways. So really interesting. So this would be really good for shotgun methods, shotgun lipidomics, proteomics, metabolomics. So we're looking forward to exploiting some of that functionality.
Hey, John,
Yeah.
we're kind of running towards the end, and I wanted just to give some folks time to answer some questions.
Okay. All right, I'll wrap it up here. This is it in action with Sam looking at the readout, and yeah, so good. That there's nothing exciting here. This is just we've identified a few projects to test, the NMT project and Tempers 2 project first off, and we're looking to integrate the academic arm and develop this workbench. Thank you. That's it for me.
This is awesome. The question already came in. Can you run this in theory in a 1536 well format?
My understanding is yes, you can. It's all the features that you would use in a typical EDC ATS 100, except that we're dispensing to the open port sampling interface rather than another plate.
Yeah, Jamie, you could use a 1536 source plate. In this case, we just use 384, but we have 1536 source plates configured.
And one other question, and I have some more I'll get to offline because this is awesome. Is what's the relationship between the acoustic dispense volume and your analytical precision? And you mentioned the sweet spot of 5 nanoliters. Is that coming from the publications or your experience, or are you just trying to cut down on how much your sample you're using?
It's primarily coming from the publications, but we know from our work to this point that we don't think it's a function of the dispense volume. We think it's a function of not being correctly aligned between the plate, the acoustic dispense plate, and the open port sampling interface or the inlet itself. So we're working on that. We did, Sam did some jiggering around, and we improved it significantly. We still have some jiggering to do.
Yeah, Jamie, if you saw the presentation from BI, the approach they took with the image, taking it in it, because essentially you have a 1 millimeter hole to hit, which is actually big in the grand scheme of things, the nanometer drop. But then what's tricky is you don't know exactly where. We assume in the center of the plate is where the droplet's detected, but we don't know, right? You can never really see that, but the alignment is one aspect of it. Because in our experience, the higher, I think that the 20 nanoliter JJ worked the best. I believe we played around with 5, 10, and 20, but we definitely got better results with 20. So it's a great question, Jamie. We're still trying to determine the relationship between the transfer volume and the peaks, but it's great. Like is the data that JJ just showed, we can quantify the actual amount, right? So, so it is, but it's a good question because there's definitely some relationship between the transfer volume.
Yep. And
well, that remains to be seen. I mean, we'll keep at it. Like it should be identical to the Artel. It should be identical to our precision. Should be that good. I mean, they're reporting 2 to 3% precision in the publications to date.
Awesome. This is great talk, John. Thank you so much for taking the time to share this with us. And can't wait to get on campus and see this in real life. So now we'll go to our final talk here, where it's going to be Bruce LeClaire, who's the head of the analytical chemistry group here at Aspire and at NCATS, me talking about advances towards developing automated purification platform.
Thank you, Jamie. Good morning, everyone. So as Jamie said, I'm today going to talk about efforts we have put forward to automate our purification platform. Currently, the Analytical Chemistry Core at NCATS, the ACC, we're responsible for the purification of internally synthesized compounds. And to perform this so that we get a efficient transfer of compounds from medicinal chemistry to compound management, we developed this centralized purification processing platform. And in this cartoon you see here, we break this down into 5 stages, coming from synthesis through purification, sample processing, registration of those compounds and then onto compound management. And you can see that what we're doing is we're funneling down to have fewer people handling the samples. And we're doing this through standardization of processes and methods. And this allows us to have 5 analytical chemists support over 40 medicinal chemists. And this data is managed through a custom, we call it smart LIMS, sample management and resource tracking. And we find that standardizing and optimizing these workflows lead to improved efficiency and increased data integrity. And a different view of this 5-stage workflow, we can see that analytical chemistry is responsible for the 3 middle stages of purification, processing, and registration. And within each of those stages, there are several steps that need to happen. And so we strive to do these 3 stages from crude reaction mixtures into stored compounds with compound management in 5 days. And this has been quite successful in that since 2009, using this workflow, DPI chemists have synthesized over 35,000 compounds. And just in the past 4 years, the ACC, we've purified over 13,000 final compounds for the compound library and over 181 different projects. And all this data is accessible through our smart LIMS, not just the analytical data, but associated metadata like barcodes of the vials and the amounts of material. And when we accomplish this through standardization of our instrumentation and methods, here on the left, you can see some standard methods we have for our purification, where we're purifying on scale of milligram to gram quantities. Particularly, we focus, my chemists typically are in the scale of probably 50 to 75 milligrams. And then we have standard methods, with acidic and basic, that we're hoping to capture a large majority of the chemotypes that are presented to us, because we are a lot of diverse projects that we do have a lot of chemotypes that come from screening. They're doing SAR around. And so then from our analytical method, we are choosing a gradient based on retention time. And these samples are then lyophilized for further processing. On the right here, you can see this is a serious liquid handler. So serious automation, a liquid handler that's been augmented with a scale, a balance, 1 and 2D barcode reading, and a vortex, and then we handle all this post-purification processing. Because that's one of the things is that we're not just focusing on purification, but processing these samples into the desired format for compound management and their efforts in managing the compounds, as well as plating for biological screening. You can see here we have some standardization in our consumables with between test tubes and barcoded vials and matrix tubes that are 1 and 2D barcoded. And this has been quite successful, but we're hitting some roadblocks and we're plateauing because a lot of this is still, there's manual steps that are involved, and we're operating in what we call islands of automation, right? So we have these distinct islands that are not interconnected, and we connect them through manual processes. So the hope is to take the centralized platform, purification platform, and automate it through more standardization to remove these manual steps and interconnect the instrumentation. And the final goal is to not only have this standalone, this automated purification platform operate in a standalone fashion, but also be a module within the larger automated chemistry laboratory. And there's always operational considerations, of course. Money is a big one, but beyond that, the expertise, looking at, do we have the in-house expertise that we need, or do we need to go out and acquire that expertise, as well as the technology? Are these things currently available, or what kind of development is needed, as well as infrastructure? Space and utilities are a big factor, and we are often constrained by space or less than ideal space. And so then, based on all this, we decided to focus on 4 development areas, being advances in our technology, management of the samples. Solvent removal is a huge concern after purification, as well as integration of these systems. So here is our idea around the automated purification platform. And as Alex and others mentioned, purification is a large bottleneck within the workflow, just because of the nature of chromatography, that is one sample at a time on a column. And so we are looking to improve this through the development what I'm calling our fluid core system, where we are incorporating an analytical LC-MS system into our preparative system. Oftentimes, you are purifying on one system and then having to take samples and walk them over to another analytical system to analyze. Or if you can do analytical analysis on your preparative system, you're choosing one or the other, which is causing delays. So we're hoping to do this in a parallel fashion so that we can purify a sample and then do analytical analysis of the fractions from the previous sample. Other things we have to look at are how do we get these samples on and off the fraction bed in an automated fashion, and then using a scheduling software to communicate between the two systems in the fraction bed so that we have the timing of this proper, because the last thing you want to do is have collisions between your arms in your system. So we are working with TCAN in the design of this fraction bed. So we are using an EVO platform from TCAN and customizing this so that we have the two arms on the system, as well as we're removing the back of the system so that we can have robotic access from the back, as well as manual access from the front as needed. And then we're working with other partners in how do we incorporate this fraction bed into the workflow. So you can see here in this flow diagram, right, we're doing reaction analysis of the crude samples, and using Versidian Analytical Studio, we will create a purification work list. Now, one of the things we're doing is that we have set methods, preparative methods based on retention time. Versidian software nicely can take retention time and based on other impurities within the sample can on the fly create a specific method for each sample rather than having standard method work lists to choose from, which the hope for this is to improve purification efficiency as well as increasing the purity. So then incorporating this fluid core system into the workflow, we have the purification that's being controlled by the Waters fraction software in operating the preparative and analytical analysis. And then after that, we have to do the fraction analysis. Again, we would use Versidian software to control this, but then post-purification of sample consolidation and processing and distribution would be happening using our smart room system. And the idea would be to interconnect all these systems into one automated purification platform. So in the previous diagram, here I'll jump back. We're seeing, approximately we have the analytical LC-MS near the fraction bed, but that doesn't necessarily have to happen. We just need a way of sampling from that bed and bringing the samples in automated fashion to the LC-MS. So yesterday, Alex talked about the lab-to-lab pneumatic tubing system, something that we're implementing, and the idea would be to connect this automated platform to the lab-to-lab. What this is, the lab-to-lab is a pneumatic sample transport system that sends barcoded 2D barcoded tubes between the labs from different areas to a central router that can then pass those samples on to LC-MS, as well as also NMR for analysis. And so you can see here that instead of walking up to a instrument, users would use a sender that then would send it onto the instrument. This affords us several advantages, being centralization of our LC-MS systems, and freeing up bench space, but also maximizing usage of the systems and being able to track the samples throughout the process. And this here can show that we can not only have chemists walking up and entering samples in a manual fashion, but we can also connect these to our automated platforms to allow integration of the instrumentation. One thing I'll show later is that this actually is an advantage in that where space is a premium, not only within the labs, but also within the individual modules, and fitting all of this instrumentation within a confined space. So moving along, talking about previously solvent removal being a large problem. And so if you remember, I had said that we are aiming to do this process in 5 days. I allocated 2 days for purification. And we currently use lyophilization, and 1 day alone is used for lyophilization. So the purification happens, within a day. We get a sample, we can purify it within that same day. But then removal of that solvent through freezing of the sample, then moving it to a bulk drying lyophilizer, and then running the process time is 20 hours. So we're eating up 1 day in just solvent removal. And one other issue with lyophilization, while it is a gold standard for solvent removal, is water, it's difficult to automate this process. And so we need to look for other ways of removing the solvent. And so that brings us to vortex evaporation. So you can see here on the right, we have two methods, either a single sample evaporation using a Biotage B10 system, or using batch evaporation with a Heddish condenser. And both of them employ rapid vortexing with heating and vacuum, and allows us to remove high boiling solvents quickly, but also that they can accommodate various vials or racks, giving us flexibility with these applications. Another advantage is that right now we currently have solvent removal and then consolidation of our samples. This allows us to combine these two steps, so we can take all those fractions, not only remove the solvent, but combine the sample into one container at the same time. And the biggest thing here is that they're automation friendly. They're already set up for automation to take commands directly from a scheduling software. So we're also looking at other solvent removal techniques, and one of these is these membrane technologies for separation of the solvents. You can quickly see that there's pure evaporation, nanofiltration, and affinity separation, and all three of them, while having some subtle differences, both use a membrane to remove one solvent and retain the other on the other side of the membrane. And based on the technique, either your material will pass through the membrane or be retained. And so we're evaluating how we can use these in automated fashion. Per evaporation is used heavily in production facilities for large scale chemical manufacture, but these systems are set up for the same process over and over again. How do we utilize these technologies with changing compound mixtures and chemotypes, moving from batch to batch? And then for affinity separation, this is actually being utilized here. And I don't know, in the Aspire program, I don't know if it's been talked about yet, but this is using as a automated alternative to a separatory funnel. So I don't know if that'll be talked about later. So we're looking at that as well for solvent removal and separation technologies. So in addition to purification, automating the actual purification, we also need to look at how do we handle these samples afterwards and getting them in a proper format that we can utilize for downstream processes. And so If you remember, it's shown you in the previous slide, the multitasker, which was doing liquid handling as well as other tasks. We have recently upgraded these systems to Omnitasker, which is a larger bed that allows us to not only you do load two process batches on at a time, but also affords us more automated processes, specifically the automated changing of gripper fingers and weigh nests, as well as the ability to aspirate and dispense slurry samples, as well as low microliter dispenses, and then this vial capper and decapper. So we're through these advances in the technology, we are removing a lot of these manual steps and having to break up the process into smaller little workflows. Additionally, we have added a microtasker that's been customized for automated sample preparation, for NMR sample preparation. And this is something that we haven't done in the past. Typically, we aliquot an NMR sample that is then passed to the medicinal chemist for them to analyze the sample. But right now, having this microtasker will allow us to actually take that sample up into the NMR tube and either potentially utilize that for automated NMR analysis, or again passing that sample off to the night chemist. But we're taking out the process of having them to make up that large sample. And in a different session, Colin Klein talked about QNMR, and we're looking at incorporating QNMR into our automated workflow, and we're looking to use this microtasker in preparing samples for that in our workflow. So how do we connect that all? So you can see here in these renderings that we are taking the Omnitasker and the microtasker and putting them together on a bench, and we're connecting them through a robotic rail arm system. So you can see here that we have this rail that runs along the back of the instrumentation, and then having a 6-axis arm that can either access the back of the Omnitasker or the side of the microtasker, as well as interconnect with other equipment, such as the Combidancer, a benchtop NMR. We have a plate sealer here, and this is a flash scanner for 2D barcode scanning for matrix racks. So by doing this, this allows us to not having to manually transfer racks between systems, between the Omnitasker, the microtasker, as well as the Combidancer. And you can also see here that based on this design, by having this rail run along the back, this allows us for safety purposes to be able to access the system in the front side for manual operation, as well as while the robotic system accesses from the back. Also, based on the swing of this arm, this allows us also along this other side of the rail to potentially put instrumentation that can interconnect. One of the things we're looking at is putting the automated, the fluid core system here, so that the 6-axis arm can reach in through the back of that fraction bed, grabbing the racks and transferring them over to the Omnitasker and to interconnect these two systems. And so I'll quickly, so our time is, I'm trying to make sure we stay on time. So here's, we're also looking at mobile robots. Yes, I'm not going to talk too much about this slide because yesterday Carlene and Jay gave a really nice talk about the use of mobile robots for biology, but they kind of highlighted a test case with analytical. You can see here that the idea of the mobile robot to also access the Omnitasker from the front side, where we manually to access that and transport racks to various systems, and this is a big benefit because we are often constrained by space. This is actually a cartoon I drew of what our space looks like with benches, and so this space is not going to change. So we have to adapt our processes and our automation to accommodate these spaces. And the other advantage is that we can look at potentially having a 24-hour work cycle. The idea of People go home for the day and the robots come out at night, right? Or how do we integrate that with, utilizing robots? Well, the chemists can go and do other tasks. All right, so I think the breakout rooms, so they're going to close, so I don't know, I have to quickly talk about, I'll skip that process, but this is what it looks like for how are we going to incorporate this into the larger automated chemistry platform. And that is where we get into constrained space. You can see here that I've in a cartoon fashion laid out schematically the purification module and the processing module, and how do we lay these components out within each module, and properly have all these processes, workflows happen, and passing it off, and also connected to a larger track system of getting crude samples in, purified compounds out. And you can see here that we're actually incorporating 2 fluid core systems, because again, purification is a large bottleneck. And while we, by going wide